Ready na. Sige.
Magandang hapon sa lahat. Good afternoon. Can you hear me? Over the Zoom at dito sa... It's on. Okay. How about on the Zoom? Can you hear me?
Yes, ma'am.
So, magandang hapon. Good afternoon. So, it's my honor to introduce our speaker for today's seminar, Dr. Patrick Cabaitan. So, Professor Patrick Cabaitan, a professor at the UP Marine Science Institute and a coral reef ecologist. So he finished his bachelor's degree in biology at University of San Carlos, Cebu City. And after graduation, he worked here at MSI under the supervisory of Dr. Edgardo Gomez at MSI. And then he took his master's degree in marine science here at UPMSI in 2002 to 2007. And then he proceeded to take his Ph.D. in marine science at University of the Ryukyus, Okinawa, Japan in 2009 and 2012. So for his work experience, so he started as research assistant here and then become a postdoctoral research assistant at De La Salle University and also a post-research assistant here at Marine Science Institute. And then in 2015, he started as assistant professor here at MSI and then become an associate and now a full professor. So through the Coral Reef Ecology Laboratory, he leads research aimed at understanding the changing dynamics among different animals living in reef ecosystem. So the broad application of his work in coral reef monitoring include the study of behavior and interaction of reef animals, bleaching, and reproductive response to disturbances such as thermal stress. His work on corals and giant clams continues the restoration efforts led by the Institute's late founder, Dr. Edgardo Gomez, which cemented UPMSI's role in marine conservation in a national scale. So without further ado, let's all welcome Dr. Patrick Cabaita.
Okay, good afternoon, everyone. Can you hear me? Is it clear? So it's my turn to share our research and future plans. And no, so I've been looking for a sexy title for my talk, but it ended up like this. Deeper understanding of coral reef ecosystems holds the key to their protection and restoration. So most research on coral reefs have been conducted to understand the changes in reef systems, the populations, different species, and more importantly, to look at what caused the changes. So hopefully with this information, we'll be able to come up with effective strategies to be able to effectively protect and restore degraded coral reefs. So here's the outline of my talk, Journey to Coral Reef Research, and then I'll share the research programs I'm involved with, giant clam conservation, coral restoration, and then finally reef monitoring. And I'll show some highlights and future plans. So my first interaction with corals and coral reefs was in USC, University of San Carlos in Cebu. So I happened uh, to help or assist in one of the projects on coral restoration, specifically coral farming. And then while at USC, University of San Carlos, I also happened to interact with Professor Montecillo. Montecillo is a Cebuano, one of the coral taxonomies in the Philippines, who've been working who work with Professor Nemenso in identifying corals. No? And then the person that brought me deeper to coral reef ecosystems is Dr. Edgardo Gomez, Professor Edgardo Gomez. So I think every talk or maybe every research will be connected with this name. No, so uh, this journey is not just my journey, but also a journey together with my students, colleagues who are now also uh, principal investigators in their own research programs. No, so we have here Van, uh, Dexter, and late Ronald Villanueva. So they are now in another meeting where I'm supposed to be attending a project monitoring for the ACR coral restoration program. So before I talk about the research programs, I would like to point out the learnings from Dr. Gomez. So the first thing I've learned from EDG was how to brush a giant clam, how to brush cages. So every student during that time has to go to Bolinao and dive with him and clean giant clam cages. And he always reminded us the advantage of having an internet and other resources because during their time, uh, they don't have internet. They write or ask for papers, articles using snail mail. No, so he always tell us everything is just in the tip of your finger. Di ba? Lahat nasa ano na, shopping, entertainment, education. And I always hear this word almost every day when he arrived in the lab. Uh, are you working hard or hardly working? And then always remind us to be creative and magtipid. Now I always remember there was a time that we were based in Bolinao, and he saw one of our setups, experimental setups. We use PVC pipes, and during his time they use nylon. And when he saw that we're using cable ties, oh, ang yaman yun na. So right now we we still make sure that we learn how to tie PVC for our experimental setups using nylon. And always reminded us to not to be shy to ask for help. No, so hindi natin kaya lahat kung ano man yung kailangang gawin. We always make sure. I mean, he always reminded us to ask help from your colleagues, other faculty, and approach them anytime. Then collaborate. And this last sentence, just choose what you can take. After my PhD, after my postdoc, I visited Sir. He's already holding yung sungkod. So he asked me, "What are you, Patrick? Are you a fish, a coral, a giant clam, or a sponge?" So he reminded me, maybe later you have to focus on one or what specific research that interests you most. And now coral reef ecology is not just about single species. It's more about communities and what drive these communities through time. No, so that's why we're my research is mostly focused on coral reef ecosystems. And last thing I'd like to emphasize this is connect with students. Uh, when I started at MSI, during the first program I'm involved with, nagyayabang siya palagi na, you see, all of the faculty here they have PhD. And he told us the secret. Why are they coming back? No, they always say that he sets the facility, he sets the environment so that the PhD students doing PhD work abroad will be enticed to come back. No, so hopefully we have now more students doing also PhD abroad. Hopefully they'll be coming back to the Philippines. So okay, so next. Now, Dr. Gomez also made sure that to connect us with other mentors. So I'm listing here Helen, taught us about complex science that's related to reef ecology. Mom Annette, because of her, most of our research is related to behavioral ecology, related to coral reproduction, which is important in the understanding of why reef communities change through time. Sir Perry, Sir Al, Sir Al, I think he's the sole, or I'm not sure, the sole coral taxonomist in the Philippines. So hopefully more of us will follow his work. So the first time I joined MSI, I approached him, Sir Al, I saw most of your skeleton, samples in your lab. Can I help and volunteer to identify those? Ang sabi niya, don't just focus on coral taxonomy. It's more interesting to work on coral taxonomy and coral reef ecology. So you have to combine, that's his advice. So now you can see in his lab, there's still lots of boxes of coral skeleton that need to be identified. Then Dr. Gomez also made sure that he invited senior students, researchers to guide us. So you have Rami Dizon, who taught us about sampling designs, experimental designs. Hildy taught us about uh, statistics and analysis. And Rene inspired me on what to do with reef fish ecology. And this is just a timeline of what I've been doing in terms of coral reef research. So from 2000 to 2009, I've been visiting reefs, or Dr. Gomez sent me to reefs that are degraded. So parang hindi siya maganda masyado kasi every time we go to the site, walang laman yung coral reef. No? So for my PhD, I decided to work on in an area na may maraming magagandang coral reef ecosystem. So I decided to stay in Okinawa and did my PhD. And then I didn't know that I'll still be going back to coral restoration. I got invited by Professor Chao Lokming to do research with him on coral restoration. And at the same time, I made sure that I also work on coral bleaching response uh, from 2012 to 2015. And now, currently, I'm working on coral and giant clam culture and reef monitoring. So in the succeeding slides, I'll be sharing the programs and what we've already accomplished and what we plan to do in the future. Now, the sequence would be giant clam conservation, coral restoration, and lastly, I'll talk about coral monitoring. Now, Dr. Gomez always mentioned that giant clams used to be everywhere in the Philippines. And he mentioned that in Bajo de Masinloc, there are fields of giant clams that won't be able to step on the substrate because they're next to each other. No, and one point I'd like to to share here is that you mentioned that if during that time, if the government only supported the program to do long-term monitoring, long-term conservation work at this site, uh, I think would still be going there very often, regularly at this time. Okay, so there are a lot of challenges to giant clam conservation. One is poaching. Even now, present time, you can still see poaching, giant clam poaching in marine protected areas. And then more recently, there's over harvesting of shells, even in terrestrial areas in the mountains. People dig in the mountain just to be able to get hold of shells. This is mainly likely because of they can be used as material for uh, sculpture. No? Then more recently, we're seeing uh, giant clams bleaching because of thermal stress events. So not just in our marines, not just in Silaki, where our giant clam ocean nursery is located, but also in other sites in the Philippines and other regions. So the history of giant clam conservation programs in the Philippines again started with Dr. Gomez. Uh, so for those of you who have not been to Silaki, so I invite you to be there. There's a homecoming event in February 22 to 23. So you'll be able to meet with alumni, ABML, Bolinao Marine Laboratory alumni, and you'll get the chance to visit the giant clam ocean nursery with tens of thousands of giant clams. So this is the history of giant clam conservation. My understanding is that during this time, 1980s, there were three marine stations that are with staff that are trained to culture giant clams. That's Bolinao, in Siliman, and one in Giwan. And the main story, why is it that Bolinao is more successful in maintaining the culture facility is because of the natural environment in Bolinao. Uh, Bolinao has a wide reef flat that uh, protect giant clams from strong wave action during typhoon. No, yung most of the broodstock in Siliman, I was told that every typhoon they get up, they roll down to the deeper areas, and eventually all their broodstock was gone because of typhoon. Now, some broodstock in Giwan, they managed to survive, but just recently, I think Odette, Typhoon Odette, most Tridacna gaigas, the big ones, more than 1 meter in shell length, are they also got affected. And then 1995 and so on, major restocking activities have been done. And more recently, we visited some of these sites and looked at how the restock clams are doing. So we both look at restock clams and natural populations of clams. So the technology to culture giant clams is already mature. I mean, we can do this. We can produce giant clams anytime in Bolinao. So we have here, we bring giant clams from Silaki to the hatchery. We spawn them, and then we collect the gametes and we rear the gametes until they become juveniles. So we let them grow in raceways in the Bolinao Marine Laboratory, and after 3 months, we transfer them to the ocean nursery, and then they stay there until they become mature, and we restock them in other sites. Now, in some sites, that with giant clams that are already mature, you can see natural uh, spawning. No? So this is the program supported by Picard that that surveyed or examined the restock clams in different sites. This is in collaboration with other universities, Western Philippine University, Davao del Norte State College, so on. Now, I would like, I'm showing this because every research program uh, we do that's been supported by Picard, we make sure that we have a partner institution. Uh, so it's a way to enhance their capacity. Now, just recently, we have a program on mesophotic coral ecosystem. We also involve Mariano Marcos State University and another university in Bohol, Holy Name University. So that way, aside from uh, sharing with them or working with them on research, they get to learn how to uh, apply for funds uh, from PICAR, from the UST, how to deal with reports, and so on and so forth. So eventually, they'll be able to do their own research, uh, do their own research. No? Okay, so what we did in that program, we conducted information dissemination and training workshop, and we did biodiversity surveys. We surveyed giant clams, and we also surveyed uh, conditions of reef in different sites. So we have around 12 sites. So in this program, Davao del Norte State College did the surveys in Mindanao. The blue ones were done by MSI. And then project two sites that's in Palawan with Western Philippine University. So what we found, most sites, or not most sites, a few of the sites we already observe giant clam recruits. So we think that these recruits come from the giant clam, Tridacna gaigas we've restocked at these sites. No? So we have here different sites with sightings of Tridacna gaigas recruits, Davao, Camiguin, Bohol, Palawan, uh, Batangas, and so on and so forth. Now, in early 1990s, the team of Ma'am Annette Nunez and Sir K. Villanoy also did a nationwide survey of giant clams. And the the density they've observed during the time is somewhat the same with the density we've recorded in this program. So hindi siya ganun kadami, except for sites that are well protected. For instance, Apo Reef, and then Calagwas. So in general, what we've noticed is that in most of the reefs we've surveyed, you can still find Tridacna corsea. This is a boring clam, not so exciting. They just get buried in the substrate. And then you have the Tridacna maxima and T. squamosa. Now, in sites that are well protected, you can find rare species of giant clams. You have Tridacna dirasa, Tridacna noe, and one of the rarest giant clam, Hippopus porcellanus. Now, aside from looking at the status of giant clam populations, we also look at whether giant clam restocking would enhance local biodiversity. So in this case, we look at whether giant clams would enhance local fish biodiversity. So you can see here, months after restocking giant clams, you can see an increase in fish communities through time. And in Silaki, we conducted another experiment to look at whether giant clam restocking had a positive effect on fish catch of fishers. In this case, fishers from Silaki. So in this experiment, we used NASA fish trap, and we deployed these in different sites: the giant clam ocean nursery, the seagrass, the lagoon environment, and in the reef. Aside from that, we conducted a fish visual census to confirm what we observed or what we captured in the uh, fish traps. So I didn't show I don't have here the figures, but we found out that there were more fish traps filled with fish in this in the giant clam ocean nursery. And when we interviewed fishers, so they also do that. I mean, they also put traps in between giant clams and catch or make use of those fish uh, for food. Now, another highlight for this conservation effort. So many of the reef or many of the restocking sites where we put giant clams have become famous tourist destination. So, for instance, here in Samal Island, this is a collaboration with different stakeholders: Davao del Norte State College, uh, People's Organization, uh, local LGU, and yeah, so those. And then 2 years into the establishment of this ecotourism activity, we already had a revenue of 2 million. That's in 2017. So this revenue continued to uh, grow higher. So you can imagine, aside from Samal, there are other sites where you can find ecotourism, where you can see restocking sites that have become a tourist destination. Now, another highlight is our restocking site in Camiguin. So we know that we established marine protected areas to protect a certain population so that they can help replenish populations in adjacent degraded reefs. So you can see here, this is Cantaan Marine Protected Area. You have here giant clams restocked inside, and you've noticed that there are some giant clam recruits outside the protected area. So that's around 2 kilometers from the marine protected area. So this is one of the evidences that if you protect a population, they would be able to uh, grow, survive, and become reproductively mature and be able to provide or replenish adjacent populations, degraded populations. But here you can see, if you just count the recruits produced from these restocked giant clams, you only have less than 10 recruits. So this restocking site is already more than, I think, 20 years. So that means this population, I mean, the recruits they produce in this area is not enough yet for what people have been doing in the past, enough for food or subsistence. No. So that means there's still a need to uh, culture giant clams and perhaps establish demonstration sites for community-based farming. This is to support restocking efforts. So this is one of the future activities that needs to be done for this program. And when when we're able to demonstrate that there are some communities are able to produce giant clams for restocking, perhaps that would help the sustainability of this conservation program. Now, aside from that, uh, there are now more sites that are capable of culturing giant clams. So not just Bolinao. So you have in Western WPU hatchery, they've produced giant clams. Semirara hatchery, Samal, and more recently we have uh, Before hatchery in Bohol. So they've Bohol has a program wherein they culture giant clams and they restock this in different marine protected areas around Bohol. So we hope to expand this effort to different sites. And since it's successful in the Philippines, so hopefully we'll be able to replicate this in other regions. So we've already deployed giant clams in Dongsha Atoll, we have in Singapore, and we have also in I forget the name of the site, uh, Borneo. Okay. Okay, from giant clam conservation, we now move to coral restoration. So just to put things into context, so coral restoration, so we have here different strategies and intervention to mitigate reef degradation. So in this setup, you can see coral restoration. You can only restore a certain area, smaller area, compared to other uh, approaches, fishery conservation, monitoring, marine protected area. But I think just like the giant clams, if you're able to grow your corals and protect them, they will become reproductively mature, and they should become source reef, uh, pro- providing larvae to other adjacent areas. So meaning their impact would be bigger if we will be able to protect them and allow them to grow faster. I mean, to grow to until they become reproductively mature. Now we have here the techniques that we've developed. In the past, we just use coral fragmentation, and more recently, we do coral sexual propagation to enhance genetic diversity, for them to become more resilient from stress, different disturbances. And since this method is expensive, meaning you have to bring or grow coral larvae in the lab and maintain a hatchery, uh, there's an effort to do coral larval reseeding. Instead of bringing larvae to the lab, we rear them in the field by just putting them in floating cages. So that's this is now one of the current efforts in the lab in MSI and in other partner institutions. And in every activity we do research, we involve the communities. So for this particular program, we're not just interested in increasing coral cover, but also learning how corals behave. So we've learned a lot. Why is it that some corals survive, some do not survive? And that would give you an idea why is it that some reef, coral reefs have a monospecific uh, community. So for instance, here, this is one of the successful coral transplantation we've done in Bolinao. We're happy we increased the coral cover, but it's monospecific. No? Another successful transplantation experiment we did, or they did. Ronald Villanueva was so excited to share this picture to me when I was still in Okinawa. They managed to increase the coral cover in the lagoon of Bolinao. This is in Binavalya. So when I visited this site, I was with Lon. Uh, what we found is just one big colony of Acropora. and there's one Drupella feeding on the in the coral. No, so that means that although you're able to increase the cover, there's still other factors that you need to examine. You need to understand why is it that some reefs are not able to recover. So I'll talk more about that in the reef monitoring slides. Okay, so just like giant clams, would say that the technology to culture and increase coral materials is relatively mature. So that means we can do this in different hatchery. So we just have to collaborate with other institutions in the Philippines. Now, what's crucial is that we need to know when corals reproduce. So we have right now a current project with ACR. We have 4 sites, Cebu, Palawan, Batangas, and Bolinao. So we're now in the 4th year. Still, we haven't done any spawning, culturing in Cebu because we do not know when the corals in Cebu spawn. And we're unlucky because when the second phase of the project started, Odette happened. So when we went to the site, you can see reef ecosystems with 90% coral cover to 0% coral cover. So partly that is maybe the reason why they are not spawning. They're still small. They're not reproductively mature. So hopefully we'll be able to observe some spawning this year. Now, aside from spawning, we noted that in some areas where there's a lot of coral spawning at the same time, spawn slick can be formed. And we tried to follow this slick, and it reached to around 3 kilometers from Anda. So there's a possibility that some of these would reach the other side of the Lingayen Gulf. No? So this is what I mentioned a while ago. Although we restore a small area of a reef, if we protect them and let them grow and become reproductively mature, the impact may be greater if they're able to provide larvae to other reef areas. Okay, so this is just a picture of how what we did with the larval reseeding experiment. So we collect fertilized eggs, put them here, and 4 to 7 days we release them to the great reefs. And here are some of the corals that some of the larvae that already grew and become reproductively mature. So right now, uh, these colonies have already produced larvae, I think, for straight 3 years already. Okay, now highlights and future efforts for coral restoration. So this is a study conducted by Sir Al, a nationwide survey of reefs. So you can see of the, I think, more than 100 reefs, only very few have high coral cover. That's 44% coral cover. Now, in Bolinao, we've been successful in creating a mobile culture system. So we managed to use Tanduyong Island, where that's where we stay. We collect larvae and do the fertilization and restocking. So perhaps in some sites where you can find areas with high coral cover that can be a source reef, uh, perhaps there's a need to develop a mobile culture system that can be used in different locations in the Philippines. Now, aside from doing that, so there's also a need to select individuals that are more resilient from stressors, thermal stress, and so on. So we do uh, selective breeding to be able to culture, individuals that are resilient. And then since we're likely maybe uh, transporting individuals in different regions. So there's also need to know whether certain populations, whether they're related to each other in a certain area. So there's a need to also map out populations that are related to each other. So we'll have a reference where up to what area can a particular population can be transferred. Okay, so that's for coral reef monitoring. Uh, if you have any questions, clarifications, just raise your hand. And okay, now we'll move to reef monitoring. So I mentioned a while ago that for 8 years, when I was with as an RA at MSI, I've been visiting to different sites, degraded reef sites. So this is all what I mean. This is the type of reef I've been encountering during the time. So for my PhD, I decided to go to a site with nice reef. So and during this time, I decided to look at or focus on looking at factors or processes why that cause or influence the change of reef communities. So currently, you have here the threats that cause reef degradation, destructive fishing, land-based runoff, ocean warming, coral bleaching. So just recently, we dive in Bolinao. We heard two dynamite blasts. So that means dynamite fishing is still uh, rampant. No, but we don't know where. And then suddenly there's a boat that passed through the speedboat. So the boatman said, "Ah, sila yun." Okay. So now the difference between these threats is that destructive fishing and land-based runoff, they are more. I mean, they're uh, they're often there. No? Ocean warming can happen like maybe every 10 years, 5 years, although right now there are times almost every year. No? So that means if corals experience these threat, they have time to recover compared to this one. No? So we have here some evidence, but sad to say it's not in the Philippines. So you can see here after the major 1998 bleaching event, you can see a drastic decline in coral cover. Then after 10 years, you can see a recovery of reef communities. And this is likely driven also by the presence of herbivorous fishes. No? I'll talk more about that in the succeeding slides. Now, here's a more recent evidence that coral communities can recover after a bleaching event. So you have here one of the reefs in Great Barrier Reef. That's 2016. Then 2020, you can see recovery of degraded communities. So how do degraded reef communities recover? So I start with sexual propagation or reproduction. So you can see here coral spawn. They release gamete bundles. Gamete bundles contain sperm and egg. Then when bundles reach the water column, they get broken down. So the sperm of one colony fertilizes the egg of other colonies. And then once egg gets fertilized, they get drifted to another reef site. Then 4 to 7 days, when they're ready to settle, they look for a hard substrate to settle onto. And then, so recruitment happens. So this is larval development, different larval, different stages of larvae through time. That's 4 to 7 days, depending on the species of corals. So this is the perfect situation. Now, you'll expect a a reef, a degraded reef, to be recovering if you find areas with juvenile corals. No? So there was a time when I visited Apo Reef. They mentioned that that area was recently affected, impacted by crown of thorns outbreak. So you can see a lot of sites with a very low coral cover. But in most of the sites, you can see small juvenile corals. So although the coral cover is very low, you can see small juvenile corals. So that means in time, uh, those particular reefs will be able to recover. Now, most of the sites or most of the reef sites in the Philippines, you can see is covered with algae or sponges, not corals. So proliferation of macroalgae is partly or mainly caused by the decline in herbivorous fishes, so caused by overfishing. And then in some sites, since there's no top predators, small fish, damsel fish, particularly farmer fish, increase in number. Now, farmer fish, they're a type of herbivorous fish also that they tend or they farm their own farms for food. So imagine if an area like this, if you can find a lot of farmer fish, uh, that particular reef will be covered with turf algae. So that means there will be no way for for a coral larvae to settle on to that reef, hindering natural recovery of coral communities or coral populations. Now, aside from overfishing, there are some sites wherein the water quality has been degraded. So, for instance, reefs near fish farms in Bolinao. So you can see. So in this site, we did some experiment. We put settlement tiles in different reefs away from a fish farm, and we noted that there's fast settlement of fleshy benthic organisms in reef site on tiles on recruitment tiles placed near a fish farm. So with that, corals won't be able to successfully settle on these tiles, again hindering recovery of uh, reef communities, degraded communities. Now, another reason why uh, many of the reefs in the Philippines are not recovering is because of uh, predation by Drupella. No? So we did an experiment. We we provided known uh, fish that are known to eat gastropods. We noted that they won't be able. They're not eating the Drupella. We provided them. So that means the top predators, the predators eating or feeding on Drupella are not anymore present in many of our reefs. So that's why Drupella uh, are proliferating. So the same with crown of thorns. Crown of thorns, the main predator of crown of thorns are a triton, a giant gastropod. So you won't be able to see them in many of our reefs. So I heard there's one site in Surigao, marine protected area in Surigao where you can still find uh, triton. No? So we're hoping to link with them, and hopefully there's a marine station or university as a hatchery in that area, and will be willing to do research on triton culture uh, and increase production of triton, and hopefully we'll be able to restock triton in different reef sites. AIMS, Australian Institute of Marine Science, has started this effort Uh, so hopefully we can also follow that program. Uh, okay. Now, some corals are naturally not palatable to Drupella. So for instance, here we did some experiment. We provided the Drupella with different species, Acropora, Cocelopora, this is Porites, and Heliopora. So the transplantation experiment I showed a while ago, uh, we managed to increase the cover of Porites cylindrica. And one of the main reasons is that Drupella do not eat Porites cylindrica. And in most of the restoration experiments we did in 2001, when you visit against those sites, they're all gone. The only species that are left are Porites and Heliopora. So perhaps one of the reasons is that they're not being eaten by Drupella. Okay, so this is just a review. So we know that corals that are bleached are able to recover, but again, because of pollution and overfishing in most of the reefs in the Philippines, you won't be able to expect them to uh, recover soon. Okay. Now, this just shows you here an example, a reef of a reef that recovered after a bleaching event. So this is because two things. One, the reefs are connected in terms of oceanographic processes, and the populations between island and island that were not affected by thermal stress are related. So there's a source of corals that allow a recovery of degraded reefs in the mainland. So hopefully we'll be able to identify these type of reefs in the Philippines. So for instance, you have here a result of surveys done by Sir Al. Perhaps the blue dots here can serve as refuge for corals from thermal stress and other disturbances. And we have to look at whether these reefs can also serve as possible source reefs. And we need to look at their reproduction. Okay. So we've been monitoring reefs. We look at coral cover. So that's one way to examine the health of reefs in the Philippines. But actually, it's not just, I mean, we have to be aware that coral cover is not enough to tell us that your reef is healthy. So for instance, some sites in Bolinao, you'll find more than 40% coral cover, but they're all covered with uh, blue coral. Again, as I mentioned, perhaps blue coral are not eaten by Drupella, so that's why they're able to proliferate. And aside from looking at coral cover, there is a need to look at the species. So at least 5 species in Bolinao that we've been using for coral transplantation 5 years, 10 years ago, you won't be able to find them now. Hydnophora rigida, uh, the Merulina, Pictinia, you won't be able to find them. So that means it's also important, aside from monitoring coral cover, there's a need to look at the species in different reef sites. So for instance, here, so you can see, if you look at them closer, this Porites looks like this particular Porites, but they are different. That's Porites monticulosa and Porites ras. So we know that identifying corals and other reef organisms is challenging. So there is really a need to enhance our capacity in coral taxonomy. So we have to work with molecular biologists to be able to identify corals to species. Now, another future effort that need to be done is to continue reef monitoring, not just coral cover, but also how they respond to thermal stress. So there are some sites, for instance, here in Bolinao, uh, you can see that there are some years corals in Balingasay bleach, but not corals in Anda. And then there are some times that only corals in Anda bleach, not in Balingasay. And often there's a different assemblage that bleach at certain years. Now, at a national scale, so we know that different sites or regions in the Philippines are exposed to different climate regime. So there's also a need to really look at reef sites in different parts of the Philippines. So how do we do that? So initially, maybe to collaborate with institutions, with marine stations. So at least uh, researchers there will have they have their own sites to monitor regularly. Okay, and then now recently we started monitoring reefs at deeper areas. So that's because of that we're able to observe rare corals at deeper reefs. So for instance, Nemenzophilia, and then Cyanarina. Mm. Then aside from corals, so we hope to also know more about biodiversity of other benthic organisms, black corals, sponge, and then gorgonians. Okay, I think this is the last slide. So these are programs that I think that need to be established or reestablished to enhance reef protection and restoration. So you have reef biodiversity assessment and monitoring, uh, examination of coral reef response to climate change, induced thermal stress, and other disturbances, enhance adaptive capacity of reef communities through research. Again, we can do this by collaborating with other institutions that happen to be nearby other reef sites in different regions in the Philippines. Then continue improving reef management and restoration. Okay, so these efforts will be successful without the help or uh, contribution or involvement of other stakeholders and funding institutions. So PCARD, ACR, DNR, and then BFAR. And lastly, so I'm ashamed I don't have a list of the students, researchers in my lab who are the main individuals who run the lab. No, so thank you everyone. So if you have questions,
Thank you, Pat. So, any questions from the audience? Who's monitoring? Let me know if there. Merong tanong? Don't be shy. Remember, don't be shy. That's why you're here for your graduate studies, so there to learn as much as you can. Anybody? Ay? Yes. May microphone ba dun? There's a microphone.
Hi. Hello? Rinig ba? Okay.
Okay.
So you mentioned earlier that there were studies or that within Bolinao you were working on increasing coral cover using monospecific. Was it intentionally monospecific transplantation, monospecific that was being done for the restoration? Why monospecific? Because di ba, wouldn't that lead it to be more vulnerable if it came to disease, if it came, especially because it was an acropora species, right, that was being used. If a typhoon hit the area, um, I believe acropora are also susceptible to coral bleaching. So if we're restoring a reef with a monospecific species, wouldn't that further down the line mean it's just like a one-time big time? It's nice and pretty now, but then one calamidad later, it's all gone again. So why monospecific?
So now days to 2024, that was done 20 maybe 2001 and so on. No, so we didn't know so much about the time, and that's the time when we started, or at least systematically testing monospecific, multi-specific, and combinations of different species. No, because if you also transplant different species, you'll expect competition. Now, some may compete, some may facilitate the growth of others, and so on and so forth.
Sorry, last one. You mentioned also earlier that there are efforts more towards restoration within the institute. I don't know if this is applicable because I've seen it more for forestry things, but the discussion within forestry is that restoration is less effective than conservation because the adult trees are more capable of providing ecosystem services than the baby trees. So which is not to say that the restoration isn't also important, but why is the focus here more on the restoration than on more large scale conservation efforts?
I mentioned I started, I first interacted with corals and coral reefs when I was in San Carlos. During the time, the thinking was management is against restoration. Restoration is against management. No, but right now the thinking is restoration should be part of management. So at least the mindset of people, researchers have already changed. So all of the efforts we've learned a lot of those things, and so that's what we're doing now. Did I answer your question?
Sure. Any other comments related? Yes, please.
Hi, I'm Prince. You mentioned earlier, kind of related to the first question, you mentioned earlier that we have been considering biophysical modeling of the coral communities. Do we have now data to suggest that some coral species are codependent with each other for their posterity? And could that be considered when they're being cultured with each other or in their spots?
Dependent, you mean species A is dependent
Yes.
sa species B.
Yes.
So I think you can see that kind of interdependence, or I'm not sure if it's intentional. For instance, in one of our experiments, if you put species A with species B, he's likely being eaten by a drupella. Now, if he's alone, likely that particular species can easily be eaten by a drupella. So I'm not sure if that's we consider that as interdependence. No, that's the only thing we've seen so far. So if we study more about how they interact at individual levels, so we may be able to find, like for instance, I mentioned competition. Right now, there are some corals when you put them next to each other, one species would cause the other corals to grow faster. So I'm not sure if that's facilitation or it's just a natural reaction because it's next to each other.
But are there efforts now to in planting multi-specific communities, culturing them? Yes.
Yeah. So you have to take a look at this as like research and more application. Now, for research, it's more controlled. We control the parameters, what to test, what to put, what to transplant. Now, in many restoration efforts, especially done by NGOs, other groups, whatever they found in the reef, whatever material is available, they use that for transplantation. But sad to say, again, after doing some transplantation work, there's no follow-up survey monitoring of what happened to those reefs or to those coral communities. So I think there has to be monitoring after a restoration effort.
Just a question about monitoring. When coral communities completely recover from bleaching, do we have evidence if they're more resistant or more vulnerable to future bleaching events?
Okay, so that's the idea. No, so not just in Philippines, there are some uh, information. For instance, in Singapore, where I did my postdoc, coral communities that are often exposed to thermal stress, they're able to uh, withstand future thermal stress events. So, for instance, we know that branching corals are more vulnerable to thermal stress than massive corals. Now, in that particular reef, uh, the branching corals did bleach after a succession of thermal stress events compared to corals in different regions. So perhaps there's that. I mean, that's one of the evidences that they can develop resistance or resilience to thermal stress. And there are current efforts in Bolinao being supported now by by ACR, and hopefully will be continued by Picard. So we were hoping to expand this effort not just in Bolinao, hope to involve other institutions because we 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 know that some reefs in different regions may behave differently in terms of response to thermal stress.
Thank you. Thank you, sir.
Yes.
Hello, sir. I have a question still regarding with restoration. So specifically on transplantation po. So, how long in general does it take to say that the transplantation is a success? And if not, how can you say that it's not? And how many percent in a 100%, let's say, does survive sa transplantation? And if a majority did not survive, do you still transplant on the same location? Ganun.
Okay. I'd like to share this information, maybe just to be so you'll be able to understand how reef corals behave. Every time you go to a reef,
Hmm.
what you're seeing is a product of factors or processes that happened to them in the past. No?
Mhm.
Unlike maybe in forest, you have a climax ecosystem or community, but in the reef, they're often affected by disturbances. So you have predation, you have typhoon almost every year. No, so that means there are some sites because of those factors they won't be able to increase their cover. Uh, Same with other corals in other regions. For instance, if you happen to visit Singapore and snorkel there, you'll find Acropora as big as this, no, or big as that, patong-patong, right turbid yung water. Now, one of the reasons is that there's no typhoon in Singapore, so they're able to grow bigger. Now, to answer your question, there are efforts where we put transplants or different groups transplant corals. So, for instance, two of the sites I've been visiting in Mactan and in South Bohol in Anda. I happened to see the reef. They're very, I mean, the cover is very high. Uh, Maybe same cover as the coral cover in Tobataha or in Apo Reef. No, that's because of coral transplantation. There are resorts that often transplant corals and they tend those, they protect them from dynamite fishing and so on. And then when Odette arrived, all of those were gone. No, So that means coral transplantation can be successful, but you also have to understand other factors that may influence the coral that you're transplanting.
Yes. Additional question din, sir, regarding sa Mactan na lang po. I am also from San Carlos, so I know that the front of the marine station is like a commercial area. And I think one of the reasons bakit nasisira doon is yung boats. Do you think na appropriate pa rin mag-transplant doon knowing that it is a commercial area?
Okay, a while ago I mentioned that restoration
Yes.
is now part of management, right? So that means before you transplant, you have to manage things. So for instance, a lot of tourist area put mooring buoys, so they tie into the mooring buoy instead of throwing anchors to minimize breakage of corals.
Yes, sir. Thank you, sir.
Okay, I think we have some questions in Zoom, so let me turn over this for the Zoom questions.
Dr. Monserpat, we have 4 questions for so far in Zoom. The first one po is from DNR1 CRFMS, from Sir Ariel Arizabal. His question is the blue coral or Helioptera a true coral?
True coral. Anong ibig? Okay. I think it's still hard kasi hindi siya soft. So I'm not sure anong ibig sabihin niya na true coral, no? So I would still say that they are still hard corals. Unlike most of the so blue coral is an octocoralia, meaning may 8 ano siya, tentacles, no? Compared to most corals, hexacoralia, divisible by 6. But blue coral, kahit octocoralia siya, they are hard. So, yeah. Depending on anong ibig niyang sabihin sa true coral.
We'll follow it up po kapag po sumagot si Zoom. We'll proceed po to the next question. From James Byhart, are there any indicators to know when corals will spawn?
Okay. Depending on the species, yung iba namumuti. So, meaning they're ready to release something. They're specifically the blue coral. They brood and then a few hours from seeing them na may mga white nag-release siya ng larvae na. Now, for some, most broadcast spawners, one of the indications is that you break a fragment and then you will see red eggs. So that means they are ready to spawn. And then if you know, or if you're already familiar with that particular coral, few minutes from spawning, you will see gametes are setting. Gamete bundles are setting. So I think para siyang may butlig-butlig, no? So I think I'm not sure if I can still share. Anyway, yun yung indication.
The next question po is from an anonymous attendee. Very nice presentation, sir. Coral restoration is expensive. In what way is this economical benefit would exceed costs in the long run? A kind of benefit that community value.
Okay. I happen to be involved in this program, Coral Reef Targeted Research. One of the main objectives of the program is to look at the cost effectiveness of restoration. No? And what we found out is really restoration is expensive. So that means better to protect reefs, manage reefs. Now, aside from, so for instance, for a particular fragment, you'll have a certain price, $2 USD dollar, $3, and so on. Now, more recent computation done by Dr. De La Cruz, larval reseeding, the piece of one piece of coral material may be a little bit expensive, depending on the number of coral material you're able to produce. So the more you'll be able to produce, the cheaper it gets, no? And then the higher survival you'll get through time, mas cheaper siya. But again, it's expensive. But what we're not able to quantify is the learnings we've obtained from doing coral restoration work. Not just us, but also fishers. Fishers would say, hindi pala bato yun. There are corals. And then they see how difficult it is to bring back corals, bring back coral cover in the reefs. And then hopefully, if we continue to do that, more fishers would get educated or get familiar with coral reef systems.
Another question for sir is from Tino Quimpo. Great talk, Pat. You mentioned sourcing dynamics is a key process to help coral reefs recover from varying disturbances. I wonder if your research or previous works have already identified these areas that could be considered hotspots to aid in recovery or conservation.
Hotspots, or meaning areas that are more resilient. Initially, I would say the data provided by Sir Al. You can see sites with blue spots. Those are sites with greater than 40% coral cover. One of those sites happened to be one of our research site. That's Apo Reef. So Apo Reef, the first time I went there, that's 2015. There are areas na almost zero yung cover. Just recently, we visited Apo Reef, so you can find again like a coral garden. So Apo Reef could be one of those sites. So in general, reefs that are offshore can potentially become like conservation hotspot, or should be prioritized in management strategy, in management or in protection.
Last question, sir, from Zoom so far is from an anonymous attendee. What is your favorite species of coral daw po?
Okay.
Slap.
My favorite, now my new favorite, I would say Sinarina or Nemensophilia. So we're excited to go back to that site and be able to quantify or study their population.
There's a manifestation to sir from Evangeline Miklat. I thank Dr. Patrick and UPMSI for the initiative of communicating the science to a wide range of audience. I am impressed to note how coral reef monitoring has greatly evolved from taxonomic studies and live coral cover measurement to a holistic approach that you presented. Thank you.
Hi, Vanji. So anyway, so any questions? So maybe a little rejoinder kasi it keeps recurring, how important is it? How expensive it is? Those of you who are studying, you are researchers, right? And that is, a lot of people need to do a lot of work, for restoration, conservation, hindi lang scientists. Pero, ang mga scientists may particular na trabaho, right? And what is that? To generate knowledge, to understand nature. Because nobody else will do that. No? Through the years, you're reading, kung wala yung mga paper na na-publish, 20, where do you start? So do not, yung sabi ni Pat, who values the learning? No, siyempre, may economic. No? But the other thing is, there are things, you work with a multi-sector, multi-agency, so you focus on the strengths of your partners. If you're a scientist and researcher, your primary responsibility is to generate new knowledge because you're being trained to do that, right? You can apply that for management and work with other people, but you need to earn your stripes. Get your paper published. They know that you've studied systematically. But there's also a lot of knowledge. Kung ayaw nyo naman, just use the best available knowledge. That's still an important skill. So the other thing that's fascinating is ecology is very complex. So you can either, ang gulo naman, maliit lang yung scale and all of that, but that's, yun eh, no? And either you see that's a problem or fantastic opportunity being in the center of marine biodiversity. Other people come here, no, para makapag-aral ng lahat ng tong confusing things na to, no, even very developed countries, and you're right here. No? So yun siguro is also one of the things that as graduate students, you have to understand, okay, there's a science for society, there's science communication. You want to be able to do that, especially to communicate the science that you do. Okay, you can read something and translate it for general public. Dapat naman. Any UP student should be able to do that. Even if you take MS 102, 101, MS1, right? Kasi ang baba-baba ng level of awareness ng Pilipino, right? Pero marine scientists, dapat mas mataas pa. You're generating new knowledge that's applicable to conservation and management. So all the study about biology or restoration, the other pitch, kasi pareho yung field namin. Why do we have to resort to restoration? It's a last resort. Kung hindi natin masira yung, we won't be having to culture things, find things. But the reality is, challenging talaga yung ating natural environment, like Patrick said, and the anthropogenic kasi kabuhayan ng mga tao yung reefs, no? So that's part of our reality. That's the social, ecological, economic context you should understand. Okay, when you do your study, you say, okay, this is a big, big picture, and what I'm trying to do bears on this. I'm making this contribution. Okay, I'm looking at this. So yun lang, no? So I just wanted, kasi paulit-ulit yung question, I think that's the benefit that we have this kind of things. So hindi kailangan basic, applied, actually you tie that together already. Okay? So any other questions? Comments?
Hello, sir. I think my question is more of a suggestion as well. Has there been any other assessment for restoration and conservation in Tawi-Tawi apart from Bungao? Kasi po there's a site or sites particularly between Sulu and the northernmost Tawi-Tawi where I think may may population of giant clams pero I think due to poaching nade-degrade siya. Actually very sad yung place kasi makikita mo talaga na patong-patong na lang yung shells tapos white na talaga siya tapos yeah parang gano'n po. I think it could be also a possible site if the situation permits in Tawi-Tawi.
Okay. Many of the collaborative work ay challenging siya kasi kung wala ng project, wala ng effort, no? But slowly we're seeing ano yung best approach to send a PhD somewhere and do the work there and expand kung anong ginagawa. Now, Richard Molil is there. He just posted a picture that he found a giant clam that we've been wanting to visit. But since siya yung malapit doon, in collaboration with Ma'am Gurley, under the Giant Clam program supported by Picard. So they're not able to see what's in there. And I know they will be able to establish management, how to protect those areas.
Okay. Anybody else? Any comments? Yes.
Sir, another question from Zoom is from Charlene Wigson. Hi, Dr. Brad. Good presentation. A few questions. What new sets of ecological questions are you personally excited now? Or what is your top advice to new students and early researchers in pursuing research?
Okay. I think I'm excited with a lot of things, complex questions. But again, complex questions require long-term effort. No, so we have when I joined MSI, some people would say, "Oh, you'll just you'll just look at how corals respond to thermal stress." So that means you'll be waiting in the lab, when there's thermal stress, right? So you have to do other conduct research, other questions that help understand how coral communities change. So that means different ecological factors uh, from community level, meaning looking at uh, diversity, cover, abundance, population level, reproduction is very interesting. How individuals interact, behavior, how they grow, and so on. Now, another level is the macroecological processes, predation, competition, herbivory. And at a more larger scale, so you have how they respond to uh, thermal stress, typhoon, and bigger environmental impacts. So that's what gets me excited. So meaning we'll continue to do that with students and again, just like Sir Al, we're hoping to do taxonomy in the future. So you're seeing his lab, a lot of mega boxes of coral skeleton. So I'm afraid that my lab is also becoming like that. So that's what gets me excited lang. And for early researchers pursuing research, just do research. And again, collaborate, uh, make connections. Now, I happen to do my postdoc in NUS, and all of the experiments, research I did there were published when I left NUS. So that means just continue the connection, the collaboration, and some of the papers when you do PhD, hindi magagawa lahat. So meaning that you will have a lot of data that you can work on in the future. Even as MS students, I still have data sa MS student ko that I have to write, and I'm excited to have time to write that paper. And currently, that's being done now in collaboration with students. So, for instance, work on increase in fish communities in giant clam restocking sites, been done with Rick Dane, Mia McGlone. So they've been they continue working on those topics. So advice, continue to collaborate, and yeah.
Yes.
Hey, sir. Good afternoon. Curious lang about the giant clam restoration. With the current success of the giant clam restoration efforts, is there a chance that the giant clams will be delisted from its current endangered status and commercialized it using the available NAP production system?
Okay. Uh, since everything started with EDG, so I'd like to talk about what his plans were and connect it with what you with your question. So community-based farming was the intention of that was to demonstrate that aside from culturing them in the lab, we're able to produce more. Fishers will be able to produce more by just rearing them in the ocean nursery. Hindi mo siya kailangan pakainin. You just put them in a hatchery and clean them out para they will have space na lumaki, no? That's the intention. And I think during the time, some institution, government agencies do not accept that idea because again, they're the ones who said that bawal magkaroon ng giant clams or mere possession of giant clams is bawal. But now recently we're happy to see that the mindset has changed. So there may be a possibility that we'll be allowing use of giant clams for other purposes, not just for conservation. As long as we're able to show them that we can track individuals. So initially, we'll start with Tridacna gaigas because we know that only a few hatchery facility can produce Tridacna gaigas. And then once we become more successful in producing colorful species, so I visited Taiwan. I told them, why are you putting them next to each other, the colorful Tridacna crocea? They said that so they won't grow. Why? Because we want to sell this to the aquarium industry. So fishers are making, they benefited this technology. Uh, Indonesia, they're also doing that effort. Now, in the Philippines, bawal, but you'll hear people that we get these from the Philippines. So that means if we're able to fix the system, able to track cultured clams, then it's possible uh, to use giant clams not just for conservation.
So, rejoinder ulit dun, ha? So, yeah, so that's tricky. The culture, same with the corals. Forever, that has been a push because they're listed under CITES. Alam niyo yun, no? Conservation of the International Trade of Endangered Species. So, it's a balance. So, definitely tama si Pat. Mas pwede siguro sa clams kasi you can track and have a certificate that it's produced in a particular hatchery. Kasi hindi naman lahat nakaka-produce. But you also have to remember that there is also the balance of conserving natural genetic diversity. And cultured animals, inherently, whether we like it, it's selected, no? So mababa talaga, we do that for sea cucumbers. It's a given. So there is going to be a balance between also protecting natural wild stocks. And hanggat maaari, if you have protected areas like that, kailangan din i-track kung saan nilalagay yung cultured, no? So that we can also track genetic diversity. We don't want that to be eroded in the Philippines kasi isa pa ring Asian. So, maraming pwedeng gawin. Yun, it's just a system, no? It's hard when you think about the system, pero operationally, sinong gagawa ng monitoring? Pwede gawin, no? So, forever na, we go back and forth with this request for... So, that's the balance. So operationally, theoretically, okay siya, pero operationally, that's a challenge. And again, here's an example where the science is very, very important. Not only for the culture, but also how you do the restoration, where you manage the restored populations. Yung hindi pa manganganak, walang problema. Giant clams, diba? Hindi naman pwede kasi they want that. Pero yung mga manganganak na may opportunity na kasi that was an issue at the GBR, if you remember. They didn't want. They said, well, this genetic pollution, no? Because you have culture, they have native big brood stock. So hindi na tinuloy yun sa GBR. No? So, pero different circumstances. So that's just another insight. Pero pwede talaga if you want to balance, conservation, maraming pwedeng gawin. Okay. Any other comments? Yes. Ito na lang.
Hello po. Regarding sa restoration, all the methods for restoration do they vary with depth? Let's say sa shallow tsaka sa mesophotic regions. And so follow-up question is, do we even know enough to even consider coral reef restoration in mesophotic regions?
Okay, quick answer. Different reefs may require different methods. No, so for instance, now you talk about mesophotic. In the past, when we dive at mesophotic depths, we can only go there. Stay there 10 minutes. Now, because and we had the training on tech diving, so we can stay 30 minutes. So there will be a limitation on what you can do in terms of activities underwater. That's including restoration. Now, most crucial, I mean, one of the crucial things to consider in restoration is how you attach corals to the substrate. Now we're seeing if you do a larval reseeding, they can naturally attach themselves to a substrate. So mas firm siya kaysa if you get a fragment and you magpokpok ka ng pako or you cement them onto a substrate. So natural attachment is better than attaching corals with epoxy and any other adhesive. So yun.
Regarding coral monitoring, coral reef monitoring. So I'm just curious when you say, for example, that in some years, coral cover changes or there is a change in, let's say, bleaching percentage or something. Do you monitor the exact same location, for example, like the exact same transect? How do you do that in coral reefs? Do you have like fixed transects, and how frequently do you do that over the course of a year, for example? Thank you.
So for reef monitoring, I'd like to share hindi lang yung approach namin but other groups also. No? So I heard that AIMS, Australian Institute of Marine Science, they go to a particular site. They don't have a permanent transect. They have GPS. They go there and then they just take video transect and then just capture pictures out of that video transect. No, in general, they every time they go there, they have data on coral cover in an area. Now, if you happen to be in a site that you can regularly visit, so for instance, Peter Edmonds, one of the expert on coral communities, I met him in Okinawa. I asked him, what did you do or what have you been doing? Why is it that you're able to track individual corals? Because in one of his publications that he's able to track juvenile to adult and then until they experience partial mortality and so on. He said that every year he went to a place for vacation, and then it just happened that there's a dive site there, and then he put a permanent plot on Transect. Now in Bolinao, we have at least 8 sites. We have permanent transects, and then we put permanent plots. Now the challenge with transects, every time we go back to the site, nawawala yung line. So that means in general na nababago yung line. But if you have a plot, like 2x2 plot, then if you take pictures of the plot now, kahit mag-move siya konti, likely you'll be able to capture corals inside that plot, so you can track them through time. Now it depends now what you want to analyze, cover or abundance or the sizes of different colonies in that particular reef.
Other questions? This is very exciting kasi mukhang maraming magtutuloy-tuloy with the coral reef research. Thank you, Pat, for giving a perspective. All of these people probably never met our national scientist, Edgardo Gomez, who is our founding director. Pero MSI, together with the seaweed study, was placed in the international because of our coral reef research. The very first international coral reef research, which put into perspective the human aspect, was done in Manila. MSC hosted that. So that placed MSI in the center of coral reef research and opened up all these opportunities for your reef researchers and all the researchers. So kayo, maraming kayong opportunities. And I really look forward, like yung giant clam, 4 decades of work, and they're teeny weeny babies. I was part of that when we first got them. They're now, we have F2, F3 already from the babies we had. No? So we'll have the mummies, the original mummies going to the BML Marijo, Heidi and Roa, Suzanne, the first people who established the giant clam cultures in Bolinao. So I can't wait to see their babies. Their apos. Okay, so anything else? So science is building, working together over the years, learning and having your networks all over the archipelago. So if you notice sa MSI, di ba, ilan sa inyo from all over the Philippines. Kailangan kayo doon. We have to do this. You know, we want more Filipino scientists as first authors for studies in the Philippines. But you need to collaborate globally kasi pwede kayong makatapat. Siguro yung isa ko pang sharing, Ed, when I was an RA, like you, I was out there, he always said, you have to think you are at par with other research assistants and graduates anywhere in the world. The science cannot be good enough for the Philippines. It has to be global standard. Ganyan. He was telling us that. Don't think, no, you have to be at par. There's no reason why you cannot be at par. Especially because we're in the center of marine biodiversity. Okay? So anything else? If not, no, Pat, thank you very much for
everyone.
sharing. No? Alam paano okay, meron certificate. Okay, so this is part of the other series of MSI's contribution to the Ocean of the Decade. We'll get announcements I think we'll have another one, no?
2 weeks.
So in 2 weeks time, so stay tuned, no? And then maybe 10, 20 years from now, kayo naman yung magkakwento sa amin. Okay, so have fun. There's merienda. Thank you for coming.
Thank you.
Thanks, Pat.